Yeah, I'm Stephen Mclaughlin. I'm a bioinformatics scientist at Medicinal Genomics. Some of the work I do is supporting Canopedia and the genomic data that we get in, the sequencing data, making sure that those workflows that process all the data are running and getting the samples up published into Canopedia and everything that kind of goes into that. So that's why I got involved in this because these were some samples that had touched my hand. So I did some analysis that I'm going to show you soon.
I'm going to put this on speaker view so we can see your slides really well.
Okay. Do you guys see my slides now?
No.
Nope.
Okay. Let me try something else. Oh, I see. I didn't finish hitting the button.
Yes.
Now you see. Okay, good. All right. Let me get this progress. I have like a bar in my way. I'm going to try to move. All right, there we go. All right, so just to get right into it, I looked at the THC synthase gene in the two skunk samples that we got from Daniel. So they're the two samples that are in Canopedia, and one is called Skunk 1 and one is called Skunk 2. And I compared it to the Weeblin Skunk 1, which I believe the story with that is that it is this, the Skunk 1 was given to Weeblin by Skunk Man Sam. And
yeah,
so
those samples came from Horticulture BV, which is David Watson's company.
So there's a paper down here that I have reference to that you can check out. So I'm calling it the Weeblin Skunk 1. And that's what I have here in this column. And so these are the genotypes at each of the 6 variants in the coding region of THC synthase that we found in the Skunk 1 sample from Hendrx Farms. And then I'm comparing it to Skunk 3 in this column. And so wild type means it's the same as our Jamaican lion reference. Homozygous means that we only see one allele at that particular locus. And wild type means that we're seeing 2 alleles at that locus. So the one that's in the, wild type means that we're seeing the same as Jamaican lion, and heterozygous means we're seeing two alleles at that locus. And so the Skunk 1 has 6 variants, and they're all heterozygous. But the Skunk 3 really looks, at least in the THC synthase gene, looks to be the same as the Weeblin Skunk 1. And there's a couple of other points of interest that I think you'll be interested in. So the Hendrx Skunk 1 is the only sample in Canopedia. So out of 459 samples that I looked at that are whole genome sequencing or are strain-seq sequencing, so I took only the samples which cover the whole THC synthase gene. Out of all of these 459, there's only one sample in Canopedia that has these 6 variants in it, and it is the Hendrx Skunk 1. All right, the other fact that you might be interested in, so since like 4 of these variants are non-synonymous and those tend to not have an effect on the coding region itself, there is, I looked at the 2 that are non-synonymous variants. And out of those 2, there's only 6 samples in Canopedia that have the 2 non-synonymous variants. So that's another fact of interest that kind of gives you a sense of how unique these strains are. Are there any questions about this slide? I'm going to get into a little bit more about how I found this information, and so we have a record of that.
I personally want to make sure I have an idea of what you're saying, because it's very much over the terms and stuff over my understanding. And anyone else in the group, this is not a space where we look down. This is a space how we learn, so we don't speak on things wrong. So please ask your questions. My question is it sounds like what you just said is that the number 3 is the one that shows the relationship to the study done in 2000, the Weeblin one. But the number one is the one that has that more rare allele. Am I correct in that?
Yes, you could say that.
Interesting.
But it's important to focus on, we're only looking here at the THC synthase gene.
Okay.
And so it's just a small slice of the genome. It's just what we had available to us that we knew was the Skunkman Sam Skunk 1 to look at. So
Right.
They gave us a limited amount of the genome to kind of look at for comparison purposes.
So that's what this data is showing us.
It's showing us the 6 variants in the THC synthase gene that are found in Hendrx Skunk 1, and then comparing what those genotypes are between Hendrx Skunk 3 and Weeblin Skunk 1. And you're right, Hendrx Skunk 3 looks like Weeblin Skunk 1 in this comparison.
Okay, Daniel.
Now I also have this other column I didn't explain is the variant frequency. And that tells you how common that individual variant is in all of the samples we have in Canopedia, the 459 I looked at where we do have complete coverage of the THC synthase gene.
And when you say in Canopedia, we were able to go into look at all the phylum, so are there other databases that we need to see if we can compare to? Potentially
we could look at phylos, but there aren't a lot of variants that we have in that database. So it's likely that some of these might be missing. So if they're not there, we can't say for sure if they're different. We just might be missing data. So yeah, I looked at these 459 because we also have a CanSNP 90 genotyping chip, but we don't have all 6 of these variants genotyped in there. So I wanted to make sure I had a large set that was as complete as possible.
All right, Ryan, Daniel, Josh, Sarah.
Yeah. I'm sorry.
Go ahead, Ryan.
I cut out right as you were starting to present, so I missed, I think, the crucial part there.
I missed a little bit at the beginning too, but I'm intrigued. I'm just soaking it all in right now.
All right.
So although heterozygous, the Hendrx Skunk 1 has that's unique that it has all of those 6 THCA synthase coding regions, and that with due diligent breeding work that gives that clone a lot of potential for exploring maybe the effect of having these THCA synthase genes, which I think we can kind of align that are unknown exactly what each one of those variants do, but the fact that all of them exist in that single clone makes it a pretty unique specimen for somebody who is exploring DNA, the effects of these different THCA synthase genes, and it could be an interesting and valuable clone. And according to Canopedia, it's statistically an anomaly so far with what data we have.
So far. All
right.
Got
so,
it.
Daniel, I think one of the things that, start to do now, once you have this information, is try to look for what sort of phenotypic differences, these plants have. And I remember talking to Marty about it early on, and he mentioned something about, the cannabinoid profile on these plants. Was one of them producing a little more THCV?
Yeah, I don't recall that. Marty is really the gatekeeper for all of the phenotype hunting work and all of the preservation work. We do play a part in it, but I credit Marty with most of the knowledge that we have in this opportunity. So we're merely the house that it lives in.
Yeah, so that could be a crumb that we could try to sniff out.
So that's something we should look at in the grow off and in the 5 growers that grow the number 1. Ryan, did you, I know you kind of got, I don't know what happened, but I think you had something to say or ask.
Well, I've got a little bit of background in genetics and like molecular genetics, not as deep as you guys, but I'm just trying to understand what you're saying. This is, you've sequenced the 1600 base pair region that they did in the Weeblen paper in these skunk plants, and you found 6 SNPs in that sequence that are different in most of the population, or at least most of your database. However, this particular plant has all 6 SNPs as per the Weeblen paper.
So yeah, in the Weeblen skunk 1 only has 1 of these, and it's homozygous. And the Hendrx skunk 1 has all 6 and they're heterozygous. So it looks like the Hendrx skunk 1 and the Weeblen skunk 1, they definitely look different looking at just this THC synthase region.
So what this has like a gene duplication in the Hendrx line? Is that what you're like? I'm trying to understand what you're saying these 6 variants are.
So it's so you have the 1 copy of the genome from the mother, 1 from the father, and so they at these 6 positions they have both alleles. So 1 copy has 1 allele, the other copy has the other.
Where are these 6 positions? Are they within that 164 base pair, 145 base pair fragment? Is that what you're saying? That these
they're in the coding region of THC synthase. So what is it, like 1600 base pairs, I think? And so the positions are given in the table here. So like, for example, 393 means that there's a mutation there that it's a synonymous mutation. So it's a proline that stays a proline. And then there's some non-synonymous mutations here. So then this is, amino acid position 63 goes from isoleucine to leucine. So that's giving you the coordinate. And then there's a genomic coordinate in this column here.
Okay.
And that's relative to the Jamaican lion genome that we have published in NCBI. Is that clear now, or?
Yeah. So essentially you've got amino acid substitutions at 6 points in the transcript.
Well, yeah, only 2 points are actually amino acid substitutions. The other 4 are synonymous mutations.
Right.
But yes, yeah.
Okay. And then I'm just trying to understand the context from a plant feeding perspective. In the Hendrix populations, there's 2 different plants. So in both of those plants, all 6 points are heterozygous?
No, only in skunk 1. And then in skunk 3, this one PALA 250 ASP is a homozygous variant that is in skunk 3 and is also in the Weeblen skunk 1.
Okay. But the other base substitutions that are in that same gene, oh no, they're both wild type. Okay.
So the Hendrix skunk 3 resembles the Weeblen skunk 1. The Hendrix skunk 1, it looks different. It has these 6 heterozygous variants.
And neither of them match what's in the Weeblen or the number 3.
They do actually have the alleles, so they all have the wild type allele. But the 6 Hendrix skunk 1 has a variant allele in it as well. So that's where the heterozygosity is coming from. You see like both copies of it.
Yeah.
And so we cover it with sequencing reads, and then half of the reads will be one allele and half will be the other when you map it back to the reference genome and call variants on it and you can look at it in a genome browser and see that there's 2 variants there at each of these positions.
Yeah, I get it. I just know the 2 populations that these things came from. The Weeblen skunk went through an inbreeding program at the University of Mississippi, breeding for isozygosity is what happened. So it's likely that that's why it's homozygous, whereas the one that went through the cannabis community wasn't, right? It wasn't bred with the intent of creating isozygous chromosomes, right? So
absolutely,
but but so really, because listen, that I'm trying to help these guys from making claims about what this genetic data shows that is beyond what it shows. Okay, that's really my intent, because the creator of this variety is a friend of mine. He's my mentor. So, like, you know, we're trying to keep the politics in line with the story. You know what I mean here? Like, that's the only reason I'm giving you the grills. I'm not trying to criticize the work. I'm just trying to truly understand what's said. So, but what you're saying is this, at least looking at the one THC acid sequence within this plant, it matches what's in the other line that we know to derive from these same seeds. Yeah.
I don't want to say the wrong thing, so honestly I didn't understand exactly the question. Maybe I just missed the specifics of what two things you mentioned.
Well, I mean, it's like, it's really interesting to know about these SNPs that cause these non-synonymous changes in the coding, but like, how does this benefit the community or the plant breeder? Right? Like, what's the real take-home message from this data?
So the real take-home message was we were just trying to see how different the Hendrx Skunk one was to the other data set. And that was really kind of the only intent here.
Okay. So it's a match. That's the story.
Well, I do have more slides. So there's okay, more.
Sorry, man, I'm not trying to cut you off.
But yeah,
so from this slide.
And thank you, Ryan and Stephen, because this is what we need, because we don't want to ruin anyone's future in this industry because we misspeak, right? And that's a big problem in this industry. So I appreciate you guys drilling down.
So what I also, and help me, you know, with this interpretation, is that in the Skunk 3, in comparison to the Skunk 1, they're both homozygous for, you know, allele 250 ASP. And that allele frequency is also present in nearly 30% of all of the Canopedia samples that you have full genome sequencing of. So it's you know, kind of like a 3 out of 10, you know, to a likelihood that it is homozygous, or is it just that it's present?
Yeah, it is a very common variant in our database. And again, we're only looking at this one gene that we had available to look at. So it's quite possible that there are several differences throughout the rest of the genome that we just don't have. We don't have access to look at those because we don't have the whole genome sequencing of the Weeblin Skunk 1. So It's just a tiny little slice of the genome that we do have at our disposal to look at.
Well, thank you. Appreciate it.
Sure. So I went into some background. This is kind of just a record keeping of the work I did and how I did it. So I took out the 4 sequences that are in that paper for Skunk 1. And these are 4 sequences that are published in NCBI. And these are the names that they have in the database. So they were 3 THCA homologs that they put in there. And then there was 1 CBD homolog that they put in the database too. And what I did was I blasted those, which is a program to just kind of look for a sequence in a large database. So I blasted them against our Jamaican lion genome. And so here are the coordinates here, the accessions and the description on the left side. Then I have the actual positions where these hit in Jamaican lion. And so the top blast hit was the gene that we have annotated in our genome as CBC synthase for this THCA2 homolog. And then this other one, which I just showed you the variant calling profile of, the one that they call THCA1 is the one that matches what we have annotated in our genome as THC synthase. And then the one that they have CBD labeled as CBD is the one that matches our CBD synthase gene that we have annotated in our Jamaican lion reference. So this is just kind of record keeping of how I did the work and how I ended up confirming that it was the area of the genome that I was interested in looking at. So I have alignments of the data in the next slides to THC synthase and CBC synthase. But since this is a type 1 plant, CBD is actually deleted. So there's no genetic data to look at there. And this is a little bit hard to see, and it's a little hard to cram all of this information into one visual slide because it is like a 1600 plus base pair region of the genome. But I'm aligning here the 2 Hendrx samples as well as the Weeblin sample in the THC synthase region, and I'm showing where these variants are. So I built a consensus sequence from the sequencing data to then align to that sequence in NCBI I pulled out that's the Weeblin skunk 1. And so in this slide, this is a little bit more than just the coding region, I think, which is why there's more than 6 variants here. And one of these positions is a no call, so that's not really a difference. So I wanted to highlight that in green so that kind of sticks out, and don't look at that. That's not important. There's missing data. It shows, it basically shows where all the differences are, and it's representing the heterozygosity as these IUPAC characters, which is just a fancy word for if it like it'll be more than one allele. So an R up here actually means that one copy is an A and one copy is a G. So that's just kind of a global view of the differences of these 3 sequences.
So the yellow is similarities and the green is no similarity.
No, actually, every other position in this genome is identical. So this is just highlighting the differences.
Okay.
Yeah, I mean, you can see over a pretty large region here, there's only a couple, really small handful of spaces where there are variants at all. So there's a lot of similarity between these.
I believe down here is the homozygous variant that I was showing you here.
Down where? Can you?
Believe it. Oh, maybe I didn't. Is that an? I think that's an A there. So I think it's right there.
There's
the only
a little like highlighter you can use on there too.
I could also break this up into multiple slides if we want to get something that has a higher resolution. I just didn't want to. I wanted to get like an overview of the whole gene. I don't know how it's coming through on the stream either. It might just look like a big blurry mess.
I can't read it. I can see the bars. Where did you highlight that?
6th row down, all the way on the right hand side.
I see the arrow now. Yeah. Okay. Where your arrow is now.
Yep.
Yeah, exactly.
Got it.
So I can try to break this up and make it, if it's something you want to share and have it be a higher resolution, we could just share the original alignment file.
It would be amazing to be able to show that with identifying where that allele is too.
Yeah, we could actually annotate it with the coding changes as well. So I can clean this up for you guys. It just let Mike know, and we'll be happy to do that.
Thank you.
And there's one more part of the story. So I did have also the CBC synthase gene to look at from the paper. One of those THC homologs hits what we have annotated in Jamaican lion as CBC synthase. And I compared the Jamaican lion mother is one of these rows in the gene. And then I have the 2 Hendrx samples. These are the RSPs. And then down here is the Weeblin skunk 1. And so there is one position in the Weeblin skunk 1 CBC synthase that has a variant not found in the other, in Jamaican lion or in the 2 Hendrx skunks. So this is a difference that I wanted to report on. It could be a fake variant, a false positive from the sequencing data. I don't know, but it's not as strong as looking at multiple variants across one gene. It's just one variant.
Sorry, that's in which plant?
This is
you're saying that
Weeblen skunk 1 has a variant in CBC synthase that's not found in the 2 Hendrx skunks.
That's not found.
Not found.
Again, these are relatively true breeding lines. So it's like any variation is not unheard of, right? It's to be expected.
Yeah, and we don't know how, long ago they, had diverged either. So
wow. I mean, cannabis is an outcrossing species. You got 40,000 plus pollen grains per anther. I mean, you're probably going to have a SNP in every fucking SNP or every single base pair throughout the genome once per generation.
Right.
So,
which gives you an idea of how, I think this is pretty good evidence then of the relationship.
And that's all we were trying to say. That's all what we hope is that there is a relationship, right?
Hey, Stephen, you didn't by chance have to look at these against Onofre and DeMeyer Mandolino's paper in 2015?
I'm sorry, what? I don't understand the question.
There's a paper, Chiara Onofre, sequence heterogeneity of cannabidiol acid synthase and THC acid synthase in cannabis sativa and its relationship with chemical phenotype. Anyway, they looked at the tertiary structure of these genes and identified, they showed with a couple of mutants why certain genes aren't churning out products, right? And I just wondered if any of these SNPs lined up with those regions of importance in this gene.
Ryan, do you have a reference to that paper you could put in the chat for us? Thank you.
I didn't look if there is any specific genomic data in there to compare this to. I could certainly add that.
Yeah, it might be interesting for shits and giggles.
Sure.
Is it an analysis, Ryan, of the genes that were analyzed in the Weeblen paper? So are the genes that they're looking at specifically related to the skunk, or are they just a different research paper that used kind of different genetic inputs and are still looking at the same region?
It's not related to the skunk, but they did. I mean, it's the plant breeder for GW Pharmaceuticals, and Sativex, one of their products, is made with skunk. I mean, the THC plant is a selection of skunk as well, so it could be the same. And they are specifically looking at CBDAS and THCAS.
Okay. If you want to provide us the reference to the paper, we could take a look and see if that has a bearing on this.
All right, and so I just have kind of the conclusions of what we talked about. One thing I didn't bring up is this additional skunk, which I think Mike might have the backstory on better than I do. The 1971 to 1974 French aficionado connection, Mike.
Yeah, it was a sample that we had gotten, I believe, from Spain, and so we sequenced that as well, and I believe they said they had some connection as well to the original. But
so I have that on the bottom here. So this is basically a screenshot of the entire THC synthase gene, looking at an IGV genome browser. So you can see the sequencing reads that are covering the region in the Hendrx skunk 1 on the top, the Hendrx skunk 3 in the middle, and then this 1971 to 1974 skunk that we'd sequenced. And one of the things that I noticed is just that the variants in that 1971 to 1974 skunk are quite different than the other 2 Hendrx ones. And this is showing that homozygous variant I told you about. in the Hendrx skunk 3, and you can see the heterozygosity in the other ones because it's represented by two colors, which represent the two alleles that are found in the sequencing reads at those positions.
Yeah, and even on the phyllo tree, the skunk 1 and skunk 3 show up right next to each other, and then the 1971-74 skunk shows up on the opposite side of the tree. So if you look across the whole genome, there's a lot more differences and variation.
So that's kind of the whole deck, just to show you the differences in your samples compared to what we found in that Weeblin paper. Are there any questions?
That's really cool. Thanks for taking the time to show this.
Oh, you're welcome. Thank you.
And Ryan, if there's anything else you think of afterwards, please feel free to reach out. We can help try to answer it. And if there's any other investigation we need to do, we can do that as well.
Yeah, it's just in the only thing I can help with is the context, right? I mean, 71, 74, skunk was still like, that's pretty early. Skunk is a 4-way hybrid, right?
Yeah.
Of four different land races. So, land races themselves are heterogeneous populations of heterozygotes. So you're going to have like potentially 8 genomes floating around in that initial, top cross 4-way, right? So it wouldn't surprise me, even if they were related, to see plants that were, that show up as unrelated, looking at those 4 markers, right? It could still be skunk, but that's the problem with what, it's kind of the secondary conversation that's comes out, really, which is kind of maybe the bigger conversation. But skunk 1 is a name, right? Like when we look at plants or we name varieties, they're usually relatively true breeding, right? And skunk 1 has been hybridized over and over and over again and released under the same name under 30, 40, 50 different seed companies. Of course, we're going to get different, things that are named the same with different genetic identities, right? It's kind of self-evident.
Right. And that doesn't even rule out folks naming, varieties because of the smell, right? And
exactly.
We're starting to learn a lot more about the thiols that are going into that.
Exactly.
And how they're produced, and that there's a bunch of different ones, and they could be produced from different mechanisms in the plant, and you can have 2 skunky smelling varieties that are not really genetically related.
Yeah, exactly.
Yeah. If there's
these things all share ancestors. So
if there's like a rubric that rates skunk to create a definition, and I think the breeder's really the only person who's qualified to name what that those reference points are to say whether or not it possesses skunk qualities or not. It seems like there's, just for the sake of this experiment, there's kind of 3 channels. There's the genetic story, there's the orative story, and then there's the story of the chemical composition, which I think, in my opinion, might include thiols. And that's only because we're at the very beginning and nobody understood what thiols were in relation to this, into this plant and this breeding project. Ryan, do you have any insights into, how the breeder might feel about those kind of 3 different, components of analyzing what we're doing here?
Well, Dave will be really interested about this work, I guarantee. It's always the thing. He's mostly interested in the oral history because he's the center of that story. He'd be interested about the chemistry, that's for sure. And then the genomic story is going to be told. It's going to tell the story. Funnily enough, Dave didn't like the skunk smell, so the guy that gave it the name, he bred away from that scent, right? Because you got to remember the context of when we were doing this. This work was being done 70s and early 80s. Like, you smelled like skunk, you're going to jail, right? So they were selecting away from that plant, even though it kept the name Skunk One. And that's much to the chagrin of today's market, where everybody wants the skunk, right? At that, they want the weed to be as loud as possible, and the parental file plays a part of that, obviously. So, yeah, I mean, I see concordance in all the stories, right? Like, I mean, it's not like there's no data here that suggests that this is the skunk. I think that it backs up that it is the skunk, right?
Yeah.
Yeah. I wish we had this data for so many different plants, guys. I mean, 1800 is great, but shit, we need this for 10,000 genomes, right?
Well, hopefully we're gonna inspire that more, right, Ryan?
Yeah. Like, well, that's an expensive challenge to get there, but man, I
would love that.
Yeah,
I'm working on it.
We have a clonal specimen that's unrelated to this project that we were gifted that carries the name Laytonville local skunk. And out of curiosity, I think it's a necessity now at this point to have that sequenced. And it's just, I think this is all really interesting stuff. And I'm really grateful for you, Ryan, and Steve and Mike and all of the medicinal genomics resources here, because these are questions that, you can ask over and over again and really just dream about the opportunity to kind of look at it. And I recognize how talented this group is, and just, I feel really grateful that we're all here having this conversation.
Now we just had 20 million to put into the research.
Next year. We're not giving up.